Hello, everyone, and welcome to today's keynote webinar titled Immunogenomic Characterization of Tumor-Reactive TIL in Lung Cancers. This webinar is a part of the Immuno-Oncology virtual event series. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located within the abstract on the left of your screen and follow the process to obtain your credits. I now present today's speaker, Kellie Smith, Associate Professor, Johns Hopkins Bloomberg-Kimmel Institute for Cancer Immunotherapy. For a complete biography on our speakers, please find it to the left of your screen. Kellie, you may now begin your presentation.
Thank you so much, Sydney, and thank you to LabRoots for inviting me to speak here today. That was a wonderful introduction, and as Sydney mentioned, I'm an Associate Professor of Oncology. I am the Director of the FEST and TCR Immunogenomics Core at Johns Hopkins, and I am very excited to present some of our work on lung cancer to you today. Here are my disclosures. I always like to start my presentations by giving a reality check of the truly grim reality of lung cancer. The truth is, 1 in 16 Americans will be diagnosed with lung cancer in their lifetime. It affects men and women equally. A lot of people don't know this, but around 15% of newly diagnosed lung cancers are in patients without a history of smoking. And around 360 Americans die of lung cancer every single day, which is the equivalent of a commercial airline jet crashing every single day. And if this was actually happening, it would be a huge public health crisis if we had an airline crashing every single day. So lung cancer is a public health crisis, and we need to treat it as such. So not only is it a significant public health problem, but it is a significantly underfunded problem as well. And what we see is lung cancer, even though it accounts for more deaths than breast, colorectal, and prostate cancers combined, it only receives 6% of federal research dollars. So it is the #1 cancer killer in this country and worldwide, but it receives nearly the least amount of funding from the federal level. So one important way that we can study lung cancer is in the immunotherapy setting. And I just want to confirm that my slides are advancing. Sorry, give me 1 second.
Sure. So Kellie, right now we see the slide that's titled The Grim Reality of Lung Cancer.
Right, and I'm on neoadjuvant. I'm on a different slide.
Okay,
sorry.
click. Do you see the button that says push to audience below the slide?
No.
Okay, how about now?
Yes.
Okay, there you go. Now go ahead and click that green button, and then just a reminder to use the next and previous buttons to advance. There we go. We can see you advancing now. Perfect.
Great. Okay, Great. Sorry about that, guys. Technical difficulties. Okay, so this was the slide that you guys, I think, were not seeing. So again, lung cancer, the #1 killer, but it only receives 6% of federal research dollars. So huge problem, but there is really not a lot of money that's being devoted to this kind of research. So one of the ways that we can study lung cancer is in the neoadjuvant setting. And this is when patients receive immunotherapy prior to their surgical resection. So if a patient presents with a resectable lung cancer, the medical oncologists and the surgeons will actually delay their surgery for a period of time so that the patient can receive immunotherapy prior to having their tumor removed. And the goal of this is to, #1, reduce the size of the tumor to increase the likelihood of curative surgery success. But #2, what we hope, and it's kind of diagrammed in this schematic here, is that it will generate systemic immunity that can seek out and destroy distant micrometastases so that it can delay or prevent recurrence of lung cancer. And this has been borne out clinically. So neoadjuvant chemo immunotherapy was approved a few years ago as standard of care treatment in the United States. And we have seen significant survival and overall survival and progression-free survival benefits when patients receive neoadjuvant chemo IO relative to chemotherapy alone. But as the laboratory immunologist, what is really exciting for me is the scientific value of the neoadjuvant setting. And what's really interesting about this setup is that it allows us to obtain biospecimens before, during, and after immunotherapy, which is not something that is routinely possible in the metastatic setting because most patients with metastatic lung cancer do not undergo on-treatment biopsies. So it's very hard to obtain on-treatment tissue. But we can do this in the neoadjuvant setting. And what's doubly important, given the interest of my lab on T-cell receptors, is that this allows a very nice foundation for using the T-cell receptor as a molecular barcode to track T-cell responses across space and across time. So across different biological compartments and then throughout the course of treatment. Now, the problem, though, is that most lung tumors actually do not respond to immune checkpoint blockade. So the response rate is in the 20 to 30% range. And our biomarkers are imperfect. And if we want to develop new biomarkers or new therapies for IO refractory tumors, we really need a better understanding of the endogenous anti-tumor immune response. And one fundamental truth of immunology, there are not many, but one of these fundamental truths is that PD-1 blockade will not work if there is not an existing anti-tumor immune response. So you can throw all the immunotherapy you want at a patient or at a tumor, but it's not going to be effective if there is not an anti-tumor immune response there to unleash. So the burning questions for my lab within my lens and my expertise is how can we detect and study these cells? What are the transcriptional programs and targets that are associated with ICB response or resistance? What other cell types do these tumor-reactive cells interact with? And what is the mechanism by which these interactions can influence anti-tumor immunity or response to immunotherapy? So we performed single-cell TCR-seq, RNA-seq on 15 tumors that were treated with neoadjuvant immunotherapy, and were able to obtain around 560,000 T cells that passed quality control. And this was published a few years ago. And one interesting thing that we found was if we looked at a pseudo-bulk gene expression level, so basically treating this as bulk RNA sequencing, we were able to separate out the tumor from adjacent normal lung on principal component analysis, but we were not able to separate out tumors based on pathologic response, which is annotated here as MPR, which is major path response, or non-MPR. So this was the indication to us that if we wanted to find some T cell intrinsic signal of response to immunotherapy, we might actually have to specifically query the T cells that are being targeted by immunotherapy, which is the tumor-reactive cells. The problem is that tumor-reactive TIL are traditionally very difficult to detect. And this was evidenced by a seminal work that was published by Evan Newell, I believe in 2019, where they queried 40 different tumors for 1,200 tumor antigen tetramers. And among those 1,200 tetramers, they only found 2 neoantigens that were reactive. And this was encouraging to me in the context of my work, because we had also been finding something similar in that it's very hard to find these cells in lung cancers. But this also was one of the first studies to suggest that tumor-reactive T cells have distinct gene transcriptional signature or expression profiles relative to bystander T cells that are irrelevant to anti-tumor immunity and are just kind of hanging out in the tumor, not doing much. But we can't really be generating 1,200 tetramers in every single study, since that's extremely laborious and expensive. So how can we make this more streamlined so that we can study large numbers of tumor-reactive T cells without having to spend all this time and money doing these cumbersome functional assays? So I want to jump back to our paper from 2021, where we performed single cell on those 15 lung cancers that were treated with immunotherapy. And at the same time, in the same issue, we co-published with Giacomo Oliveira and Kathy Wu on their lung cancer data, where they also identified tumor-reactive cells. And if you look on the left, in our lung cancer data, we have mana-specific T cells, which are neoantigen-specific T cells, that are shown in red, which are those cells that are recognizing the tumor. And then we also have flu-specific T cells in blue and EBV-specific T cells in purple. And most of the neoantigen-specific T cells actually localize to a tissue-resident memory, or TRM cluster. On the right, Giacomo also identified T cells that had different antigen specificities. And most of his tumor-reactive cells localized to what he called a terminally exhausted T cell cluster. But if you actually look at these clusters, they did have similar gene expression. So we called them different things. But if we look at the genes that are actually being expressed by the cells in these clusters, some of the genes were similar. So this brought up the question to us of, are there transcriptional signatures among tumor-reactive TIL from different cancer types? So is there some kind of shared transcriptional signature that we can generate in a pan-cancer fashion to better study these cells in higher numbers than what we'd be able to identify with functional assays? So we performed an integrated clustering where we obtained Giacomo's data, and we ran everything through the same pre-processing pipelines and integrated these cells into a single UMAP. And indeed, when we look at the tumor-reactive cells, which are shown in red, so the neoantigens are in red, the melanoma-associated antigen TIL are in green, which were identified in Giacomo's melanoma data set. These cells actually co-localize to identical regions of the UMAP. So again, we were calling them different things, but when we actually look at them on the same playing field, the neoantigen or the neoadjuvant treated TIL from lung cancers localized to the same region of the UMAP as the tumor-reactive TIL from melanomas that were untreated, right? So we have 2 different tumor types, and we have 2 different treatment settings. So this was very encouraging to us. And what we found was that there were 3 main genes that provided the foundation for this shared transcriptional signature. And those genes were, it was high expression of CXCL13, high expression of CD39, which was previously identified by Evan Newell, and then low expression of the IL-7 receptor. So we decided to develop a gene signature based on these 3 genes, and to test how it performed against currently available gene signatures. So Zhen Zhang, who was a postdoctoral fellow and is now junior faculty in my research group, she developed a discovery test and validation pipeline for this score, which we are now calling MANA score. So we trained the score on the melanoma data. And then we were able to test and validate on various publicly available data sets that have published transcriptional signatures of validated tumor-reactive T cells. And many of you are probably familiar with the Rosenberg neo-TCR signature. So we first compared our MANA score test data against the neo-TCR8 gene signature, as well as CXCL13. And we did this for imputed and non-imputed data, just to make sure we're covering all our bases, because there are some questions or concerns in the field about placing too much emphasis on imputed data. So we wanted to address those issues by comparing against both imputed and non-imputed MANA score. As well as CXCL13, which has also been suggested as a marker of tumor-reactive T cells. So as you can see with this left ROC curve here, MANA score outperforms the currently used neo-TCR8, and it also outperforms in the validation data as well, which is shown on the right. So just to emphasize, this 3-gene signature outperforms neo-TCR8, which is a 240 gene signature. So this is really exciting for us because, the performance is great, but it's a much more parsimonious gene signature. We only have 3 genes as opposed to 240. So that really opens the door for implementation into multispectral IF and other kind of spatial analyses. One really cool thing, too, that we're able to address with MANA score is the inter and intra-patient heterogeneity of transcriptional signatures. So for those of you who are in this realm and have studied these kinds of data sets, you will know that if you look within individual T cell clones, so these are cells that share the same T cell receptor and therefore they recognize the same antigen, even if you're getting all of these cells from the same microenvironment, there is a wide range of transcriptional programs that you will see in these cells. So what I'm showing here is the overall MANA score, which is taken as the square root of the imputed MANA score squared plus the non-imputed MANA score squared. And this was, very fancy computational approaches that were developed by Zhen in collaboration with Hongkai Ji. But what you will see here is that, so on the y-axis, you have this range of MANA scores. On the x-axis, you have each of your CD8 clones that were identified among the tumor-infiltrating lymphocytes. And you have this wide range of MANA scores within individual clones. You also have a range of MANA scores within patients. So MD01005 and MD01010 are 2 of the patients from our neoadjuvant study. And you can see very different MANA scores between these 2 patients. But what's interesting is we validated tumor-reactive cells in both of these patients. So we know that both of them have tumor-reactive cells, even though their MANA scores are very different and very diverse within each patient. So how can we develop a patient-specific threshold that can capture or account for these differences? And that's one really unique thing about our MANA score is rather than having a set threshold or an absolute cutoff for what we're calling MANA score high versus MANA score low, we actually have patient-specific thresholds that we set. And these thresholds are generated by looking at the imputed and non-imputed values and taking the upper right quadrant of cells when looking at the non-imputed and imputed MANA score. So this, again, allows us to really tailor this to each individual patient. And it's also good because not in every single patient will you detect MANA score high cells. Because we also have a threshold to the number of cells within a clone that have to be MANA score high. So this really is, in my opinion, a really creative way that Zen has figured out how to account for these issues that we've all kind of acknowledged in the field, but we haven't really developed a solution to. We then wanted to compare our MANA score to functionally validated MANA-specific TIL. So these are TIL, again, from this neoadjuvant study that we identified functionally. I'm showing the overlay of these cells on the UMAP. So again, these red cells are all localized to this tissue-resident memory cluster. And when we look at our putative tumor-reactive cells as identified by MANA score, so these are cells with a TCR that falls within that upper right quadrant, we see nearly identical overlay of the putative tumor-reactive cells, or PTRCs, with our functionally validated neoantigen-specific TIL. This was the first indication to us that we were actually onto something, and that we're actually, we're actually having a high specificity in terms of our score being able to predict the identity of these cells. We then obtained another publicly available data set from oral cancers. And again, when we look at the validated tumor-reactive TIL, which are shown in the left on red, and then we overlay our MANA score, we see a very close overlay of our score with the validated T cells from that cohort. And then again, from another publicly available data set, this was from the Steve Rosenberg study. Again, validated TIL on the left, MANA score on the right, and we see very nice concordance between these two. So we next wanted to test if MANA score is able to identify cells that recognize other classes of tumor antigens. So we've captured neoantigens, we've captured melanoma-associated antigens. This oral cancer data set that I'm showing here on the left had endogenous retrovirus-specific T cells, so we're able to capture, some dark antigens. Our next question was, can we capture T cells that recognize viral oncoproteins? And really, the best model for this is Merkel cell carcinoma. So we generated a CD8 positive single cell data set from virus positive Merkel cell carcinomas. And we implemented site-seq technology with multimers specific for known immunodominant epitopes from the Merkel cell polyoma virus. So these cancers have very, very low mutational burden and are primarily driven by this Merkel cell polyoma virus. And it has been documented that most of the T cell responses are to the large T antigen of the Merkel cell polyoma virus. So this was a very nice controlled setting where we actually know what the likely antigen is. And we were able to, again, use these multimers in a site-seq type of way by coupling it with single cell to identify the epitope-specific T cells within the single cell data. And we were able to identify a little over 4,400 Merkel cell polyoma virus-specific TIL based on the multimer approach. And our MANA score really nicely, again, overlays with where these cells are on the UMAP. And these are all tissue-resident memory clusters. So not only are we seeing conserved transcriptional signatures, but they're also, as expected, localizing to the same types of clusters and exhibiting the same types of features as T cells that recognize other classes of tumor antigens. And what was really nice, if you look at the Venn diagram on the right here, is we were able to capture greater than 99% of the multimer positive cells when we used our MANA score. So there were only 42 cells and 6 clones that we were not able to capture by Manascore. Now, there were around 4,000 cells that were identified as PTRC by Manascore that were not captured by the multimers. So here we're talking about a specificity issue, right? One thing that we're doing to account for this is instead of testing 5 immunogenic antigens from the Merkel cell polyomavirus, we are actually generating libraries spanning the entire large T antigen, so that we can capture every possible antigen that is immunogenic in these patients, so that we can increase the number of PTRC that are validated functionally. So this is all in the works now, so that we can get a better idea of the true sensitivity and specificity of our score. And I know that this says impress here, but this paper actually was recently published in Nature Communications. So I encourage everyone to go check it out. So all of this development is great. So how do we actually apply this in the real world and apply it practically to discover new biology? So the lowest hanging fruit for us was just to look at if Manascore is somehow associated with clinical outcome. And indeed, if we look at the proportion of putative tumor-reactive cells within the tumor, it is higher in responding tumors relative to non-responding tumors. Now, it isn't the most striking difference in the world. You know, we have a p-value of 0.047, but these are human data in a clinical trial, in a small clinical trial. So we were actually pretty pleased and excited with this somewhat preliminary finding. And when we look at the proportion of PTRC as a function of percent residual viable tumor, so looking at this as a continuous variable, we do see this negative association between the frequency of these putative tumor-reactive cells and the percent of viable tumor at the time of surgery. The next thing was to start to understand what are some of the key functional differences between tumor-reactive cells from responding and non-responding patients. So we performed just a basic differential gene expression of putative tumor-reactive cells in responders and non-responders. And on the left, you'll see that granzyme K was the most highly upregulated gene in tumor-reactive TIL from responding tumors, whereas granulysin, granzyme A, interferon gamma, granzyme B were all upregulated in the non-responding tumors. And this was really interesting to us because we often associate, granulysin, gamma, granzyme B with an effector T cell response. But we're actually not really seeing that in the context of our data. And I'm going to touch on that in just a few slides. So this is all good and well, but neoadjuvant immunotherapy by itself is actually not the standard of care, right? This was as part of a phase 2 clinical trial that we performed at Johns Hopkins. But in the real world, immunotherapy, in 80% of patients, immunotherapy is combined with chemotherapy. And to date, we really have no idea what is the effect on T cells when you combine immunotherapy with chemotherapy. Because we traditionally think of chemotherapy as being lympholytic, it really, quite frankly, made no sense to me and many other immunologists when they started combining these two agents together. It almost seems like you're going to shoot yourself in the foot if you're combining a T cell activating agent with something that traditionally kills T cells. But clinically, that we did not, our concerns were actually assuaged because clinically, it seems to be very effective. So we've initiated this very large study led by Sydney Connor, who was a Ph.D. student and is now a postdoc in my lab, where we are able to directly compare tumors from patients who went right to surgery without any therapy, who received anti-PD-1 alone, and who received anti-PD-1 plus chemo. And this is a very unique cohort because these patients were all treated within the same clinical trial. And surprisingly, the The clinical trials of Nivo alone or anti-PD-1 alone versus anti-PD-1 plus chemo have never been performed and likely will not ever be performed. So this direct comparison really is only available to us because we did this trial, and we have this direct comparison of these two different treatment arms. So Sydney performed single-cell TCR-seq, RNA-seq on these three cohorts that I've listed here, and generated this integrated UMAP of almost 700,000 CD8 positive T cells. And the very first thing that we wanted to check was if the chemotherapy arm was ablating the proliferating T cell cluster. Because chemotherapy targets proliferating cells, you would think that proliferating T cells would be eliminated when you combine IO with chemo. And just to kind of orient you, the proliferating cluster is the top right pink cluster shown on the UMAP. And what we saw is that actually that is not the case. We are not seeing a complete elimination of this proliferating cluster. If you look at the UMAP on the right, which is showing the ICB plus chemo treatment arm, cluster 11, which is proliferating, is actually alive and well. So this was somewhat shocking and, very counter to what we would expect, but that's science and that's what makes it interesting. So we again performed this pseudo-bulk principal component analysis, and we were able to separate out the treatment naive versus neoadjuvant treated TIL. So if we just compare all tumors that received treatment, regardless of what it was, versus untreated TIL that just went, these patients went right to surgical resection, we are able to separate them at a pseudo-bulk level by principal component analysis. But what was very interesting is if we directly compare the PD-1 alone versus PD-1 plus chemo, we actually don't see a separation anymore. So this indicated to us that the effects introduced by chemotherapy were much more subtle. And again, we're going to have to dig a little bit deeper into the different T cell subsets and putative tumor-reactive cells to see if we can start to see some differences. So we applied MANA score to this integrated data set and saw no difference in the average MANA score between our treated cohorts. And we also didn't see a difference in the frequency of putative tumor-reactive cells from the resected tumor. So this is just to say that the addition of chemotherapy is not negatively or positively impacting the infiltration of tumor-reactive cells. And we also see when we overlay putative tumor-reactive cells onto the UMAP, they really localize to identical regions on the UMAP. So again, global differences in transcriptional programming are not what's driving this. We need to dig deeper. So we performed differential gene expression between the putative tumor-reactive cells in our two groups. And what I'm showing here is a volcano plot, and we're showing the genes that are upregulated in the PD-1 alone on the left and PD-1 plus chemo on the right. And what you'll notice is our favorite genes, again, are popping up. Granzyme K is very highly expressed in the PTRCs from the combination treatment, whereas granulysin, granzyme B, and perforin are upregulated in PTRCs treated with immunotherapy alone. So these are the same genes that were differential when we looked at responders versus non-responders in our original immunotherapy alone cohort. So what this indicates to us is that the tumors treated with Nivo and chemo combined are actually looking more like our responders, whereas the tumors treated with Nivo alone are looking more like our non-responders. And what's really cool too is that this is decoupling granzymes, which have traditionally kind of all been lumped together. So a lot of us for many years just looked at granzymes as kind of all, in one same basket. But what we're seeing is this consistent decoupling of granzyme B and granzyme K, which was very interesting to us. So we wanted to ask, could this decoupling be indicative of a separation between exhaustion and activation, right? So a lot of the genes that we think of as exhaustion markers are also really just markers of T cell activation. So we applied some publicly available transcriptional signatures to our putative tumor-reactive cells from our 2 different cohorts. And we saw that there was no difference in expression of checkpoint signatures. So this is a gene signature comprised of PD1, CTLA4, TIM3, LAG3, all the usual suspects. There's no difference. But what we do see is this decoupling of exhaustion and cytotoxicity. So the PD1 plus chemo PTRC have much lower expression of an exhaustion signature, but higher expression of a cytotoxicity signature. So a lot of folks, if you do this kind of work and you work with these signatures, you will see many times that exhaustion and cytotoxicity tend to go hand in hand because a lot of the genes are conserved across these two. And it becomes very hard to kind of separate out T cells that are undergoing cytotoxic functions versus T cells that are on their way to exhaustion. But we are able to separate this out, and it seems like the addition of chemotherapy to PD-1 blockade is actually promoting a differentiation into more of a cytotoxic phenotype and less of an exhausted functional program. So we next wanted to assess if any of these treatments are influencing the interaction of putative tumor-reactive cells with other cell types in the TME. And because we've done a lot of work with regulatory T cells and we had a lot of data already, this was one of the first places that we decided to look. So a few years ago, we published on the transcriptional programs of regulatory T cells in these same patients treated with neoadjuvant immunotherapy. And we found this cluster that we call activated 1 that had high expression of OX40, and it was highly immunosuppressive ex vivo and in vitro, much more so than other Treg subsets. So we know all Tregs are somewhat immunosuppressive at different stages, but these Tregs were actually exceptional in their ability to suppress T cell proliferation. So we thought maybe we should query some of the ligands of these markers. And this is particularly important because in this data set, these activated Tregs were associated with response to immunotherapy. So we see that non-responding tumors had a higher proportion of these Tregs in the tumor relative to responding tumors. And when we queried our putative tumor-reactive cells, those from non-responding tumors had much higher levels of the ligand for OX40, which is OX40 ligand. And this actually indicated the potential for direct interaction of our tumor-reactive T cells with this highly immunosuppressive subset of regulatory T cells. What was also interesting is that we had this very small population of Tregs that had features consistent with a T helper type of cell. So they express interferon gamma ex vivo. They had high levels of T-bet. They had somewhat lower levels of FOXP3, but they were still Tregs. And these cells were higher in responding tumors. So we have these two dichotomous subsets of regulatory T cells that are differentially associated with response and have the potential to influence our tumor-reactive CD8 T cells. But this is only something that can be inferred by single cell data. It really has to be resolved spatially because some of these, like the OX40 ligand interaction, is a contact dependent mechanism. So it's only if these cells are directly interacting where this association would be relevant. So our next question was, can we analyze this spatially? So we performed spatial transcriptomics on some of these cases, the same patients treated with neoadjuvant IO or IO plus chemo. And really the cool thing about the neoadjuvant platform is that it allows us to study mixed treatment effects within the same resection specimen. So within the same tumor, you can have regions of immune-mediated regression and regions where there seems to be no treatment effect at all. And that's exactly what we're capturing here. So in the blue, we see a tumor nest that had no treatment effect. And then in green, we have an area of immune-mediated regression where there used to be a tumor, but now there are features consistent with regression. So we performed spatial on these regions, and the spatial transcriptomics validated the pathological assessments, where we have the area of tumor in the top left, and then we have an area of lymphocyte infiltration on the right. So Zen developed a modified version of MANASCORE to be applied spatially, which I'm not going to go into today, but it involved a lot of optimization and back and forth and work and things like that. But when she applied a spatial MANASCORE, we found that there are what we would call putative tumor-reactive cells in both of these regions. Although in the area where there's no regression on the top left, it really looks like these PTRCs are restricted to the margins of the tumor and really didn't infiltrate that well. When we overlay our activated Treg signature, so these are our Treg that were highly immunosuppressive, they are largely localized to the tumor nest that had no regression and no treatment effect. Whereas our Th1-like Tregs are kind of seen in both of these regions, but really you can see them in this area of immune-mediated regression. So we then wanted to look at the distance between our putative tumor-reactive cells and these different regulatory T cell subsets. So what I'm showing here is on the y-axis, we have the nearest distance to a MANASCORE high cell for our tumor nest and our area of regression. So a smaller distance means the cells are closer together. And what we see is that in our area of regression, our activated Tregs are much farther away from our tumor-reactive cells. Whereas in our tumor nest, these cells are much closer, indicating the potential for more of this immunosuppression in the tumor nest that had no evidence of treatment effect. Whereas if we look at our Th1-like Tregs, we see the opposite. So these Tregs that express interferon gamma, they express TBET, they have all the features of a helper cell that would be good for anti-tumor immunity. And these cells are closer to our MANASCORE high cells in the area of regression relative to the tumor nest. So this is all kind of a work in progress for us at present, and this is actively ongoing. But this was very encouraging to us that our signals that we're getting from our single cell data might actually bear out spatially and might actually be biologically and clinically relevant in a spatial capacity. But the remaining question is, what is going on with this granzyme K and granzyme B stuff, right? So we have our interaction between our PTRCs and our different Tregs, but what is the role of granzyme K and granzyme B in this kind of spatial milieu? So when we look at the spatial localization of these genes, granzyme B is on the left and granzyme K is on the right. We see that granzyme B is largely in these CD8 T cells at the site of the tumor nest that does not have regression. Whereas our granzyme K expressing cells are really largely in this area of immune-mediated regression. So we hypothesize that granzyme K marks a sustainable, almost stem-like T cell that has been shown time and again to be important and necessary for anti-tumor immunity, whereas we think granzyme B cells are really indicative of exhaustion. And granzyme B has been a very good marker for studying tumor-reactive cells, and they are tumor-reactive, but it seems like they possibly aren't functioning as well as they should, or as well as they did probably previously. So we're currently studying these, and we're finding that granzyme K and granzyme B tend to be mutually exclusive, and they really mark very distinct T cell subsets. So in conclusion, hopefully what I've been able to show to you today is that we are able to identify tumor-reactive TIL with high precision with only 3 genes that accounts for inter and intra-patient heterogeneity. we are able to capture CD8 T cells that recognize different types of tumor antigens. So neoantigens, tumor-associated antigens, endogenous retroviruses, viral oncoproteins, et cetera. We can use these genes to discover new biology of tumor-reactive TIL because we're no longer restricted to the few number of cells that we identify by functional assays. We have a much larger pool of T cells that we're able to study. And currently, our ongoing questions are, what is going on with granzyme K? That is kind of the burning question for me right now. And are Th1-like Tregs really a good thing for ICB response? So what is promoting the generation of these Th1-like Tregs? Are they influencing ICB response? And how are they influencing ICB response? And then we've shown that Manascore can be implemented spatially. And again, I put this on there twice. What is going on with granzyme K? Because I think it's one of the most interesting questions that we've been able to get out of this data. So with that, I would like to thank all of the members of my lab, especially Zen and Sydney, who did most of the work that I presented here today, our medical oncology collaborators who led all of these clinical trials, Elsa Anagnastou, especially from our cancer genetics, Honkai Ji from biostatistics, who is really our leader for all of our computational work, Janice Taub from pathology, our funding, and of course, the patients and their families. And with that, I would be happy to take any questions. Thank you so much.
All right, perfect. Thank you, Kellie, for your very informative presentation. We will now jump into the Q&A portion of the webinar. So if you have a question that you would like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. All right, let's go ahead and get started here. Okay, so the first question that we have says, are you exploring any ways to therapeutically harness the differential TREG subsets you discussed?
Yes, yeah. So I can go back if folks can still see my screen. I would love to go back to discuss that. So if you recall, we have these Treg subsets that one of them is highly activated and immunosuppressive and expresses OX40. And one of them is Th1-like. We are in collaboration with Jamie Spangler here at Johns Hopkins. She is a wonderful immunoengineer and protein engineer who is developing a IL-2 mutein that specifically binds to CD25 on regulatory T cells, but does not activate CD25. And it also binds to OX40. So this is a mechanism by which we can specifically inhibit the OX40 high expressing regulatory T cells while sparing the other Tregs that are in the patient. And this is especially cool from my standpoint, because if you think about treatments like anti-CTLA-4, the reason they are so toxic is because you really just deplete all of your regulatory T cells. And a lot of these are important for staving off autoimmunity. So that's why you get a lot of autoimmune toxicities with these immunotherapy agents. But this is a mechanism by which we could potentially reduce some of that, because you're really only getting rid of the worst players. You're not getting rid of all of them. So this is something that I am very excited about and that we're pursuing right now.
Perfect. Thank you. Okay, it looks like we have a couple more here. The next question is, what do you think is the relationship of granzyme K to the other granzymes?
Yeah, I think, granzyme K, traditionally we thought of as a marker of an early effector cell, and we think that very well may be the case. When we're doing some trajectory analyses in pseudotime, what we're seeing is that granzyme K tends to pop up earlier in the pseudotime than granzyme B. So we think that as a T cell transitions from an effector T cell to more of an exhausted T cell, it will actually lose granzyme K and upregulate granzyme B.
All right, wonderful. Okay, it looks like we have one more question here. Do you think there is a role for Manascore in patient care / decision making?
Yes. As I kind of alluded to, because the MANA score is only a 3-gene signature, it does make it compatible with multispectral immunofluorescence. So we are developing panels right now to implement MANA score spatially. And this is something that in theory could be used in the clinic to guide decision making in terms of predicting response to immunotherapy or in identifying candidate therapies if and when they become available.
All right, wonderful. Thank you. Thank you again, Kellie Smith, for your time today and for your very important research. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webinar can be viewed on demand for 1 year until April 30th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, goodbye.